So dear students, in this session we will be discussing mainly about the basics in microscopy. And before moving into it, let me introduce you to myself. I am Dr. Sasango Sandilya, HOD Department of Microbiology of the Assam Kaziranga University. So today we will be mainly discussing about the introduction of microscopy and its history, followed by the variables that are being used in microscopy, and thirdly, that means lastly, we will be discussing about different types and uses of some common microscope that are generally used in research laboratories. Now, while introducing this microscopy, first of all, we would like to discuss about microscope, the term microscope. So it has been divided into two halves. One is micron and the other one is scopios. So micron means small and scopios means to look. Microscope is nothing but an instrument with the help of which we can visualize different kind of objects which cannot be visualized by our naked eyes. And microscopy is the science that deals with this microscopy. So the science of investigating small objects using an instrument is known as microscopy. Now let us go through the history or the historical background of microscopy. So in 1590, Hans Janssen and his son Zacharias Janssen, two persons, they were involved in the development of the first microscope. In 1609, another scientist called Galileo Galilei have developed the compound microscope. Thereafter in 1620, it was noticed that one Dutchman called Christian Huygens, he has developed a two-lens ocular system. That means the microscope in which two eyepiece lenses were available and which was later on chromatically corrected. Now see this particular microscope which was developed by Anton van Leeuwenhoek. This was a very primitive kind of a microscope that was being used initially. Later on, another scientist called Robert Hooke have developed a microscope which is, you can say it's a sophisticated kind of microscope as compared to that of Leeuwenhoek. But nowadays, if you go through different kind of laboratories or different institutions where microscopes are available, scientific research institutes mainly, there you will be able to visualize different kind of very, very sophisticated microscopes. Now, before discussing about what is microscope and what is microscopy or what is the science that is behind microscopy, we will have to learn about or we will have to know about the variables used in microscopy. So there are different kind of variables which nowadays you frequently come across these terminologies, mainly the magnification. So if you see magnification, it means the degree of enlargement. That means up to which level this a particular object can be enlarged. Okay. So number of times the length, breadth or diameter of an object is multiplied. So the number of times length of an object, breadth of an object as well as diameter of an object, if it is multiplied, then it will be termed as magnification. It depends upon certain factors. Number one is the optical tube length. Number two is the focal length of objective, and third one is the magnifying power of eyepiece lens. So these are the three major factors which are responsible for a better quality magnified image that you will be able to visualize. Now if we come across total magnification, or if we want to define what is total magnification of a particular microscope, then what we need to do is we'll have to multiply the magnification of the eyepiece lens as well as magnification of the objective lens. Next one is resolution. I'm sure that all of you have come across this terminology called resolution. But if somebody asks you or ask you to define resolution, then it sometimes becomes difficult for us to define what is resolution, right? So it is a very simple terminology where it is nothing but an ability to reveal closely adjacent structural details as separate and distinct. Now let us see how this can be ascertained. Like for example, here you can see, suppose these are two different objects. Now if these are two different objects, you have to, if you are looking it from a long distance, then it may appear like both those objects may appear to be same for you if you are looking it from a distance. Now if you go close to that particular object, then only you will be able to visualize that these are two different objects. Okay, now that particular difference will be provided to you by this resolution, okay, by this ability of the microscope, which will reveal closely adjacent structural details separate and distinct. That means you will be able to visualize, okay, yes, these two objects are now actually these two are separate and distinct, okay, but it is not the same that I was thinking when I was visualizing it from a far far distance. Okay, so then there is another terminology called limit of resolution. Limit of resolution is the minimum distance between two visible bodies at which they can be seen as separate and not in contact with each other. So just now I have told you about two different objects, right? So if these are two different objects, now limit of resolution will be the distance between these two objects. Okay, as we have seen these two objects to be separate as well as these two objects to be distinct from each other. So now if you calculate the distance between these two objects, that will clarify your, or that will be your limit of resolution. Okay, the minimum distance between two visible bodies at which they can be seen as separate and not in contact with each other. Now mathematically it is defined as LR is equal to 0.61, which is a constant, into W, which signifies wavelength, by NA. NA means numerical aperture. Now what is NA, that we will discuss in the next slide. Okay, now numerical aperture. What is numerical aperture? Now let us see, ratio of diameter of lens to its focal length. Okay, ratio of the diameter of a lens. This is, suppose if this is the lens, it has got a particular diameter. So ratio of the diameter of the lens to its focal length, this is the focal length. Okay, so from here the ratio, if we calculate the ratio of the diameter of the lens to its focal length, then it will be your numerical aperture. Okay, now mathematically it is written, or mathematically it is expressed as NA is equal to n sin theta by 2, where n is your refractive index. So where n is equal to your refractive index, and theta, this theta is your angle of aperture. Okay, then comes next variable, which is definition. Now a capacity of an objective to render outline of the image of an object clear and distinct. Now what does this mean actually? For example, suppose there is an object. Suppose this is an object. Now how can we say that this is an object? It can be said only and only if you have got an outline of it. If there is no outline of any object, then you cannot define that this is some kind of an object, right? So in that case, what we will see the outline. So here this definition means the capacity of an objective, that means the capacity of an objective lens which will render an outline of the image. Okay, that means which will help you to see an outline of the image of an object, and that to be very much clear and distinct. If the objective lens is providing you with a clear outline of an object, then this will signify this particular variable is termed as definition. I hope this will be clear for you. Now next variable, which is the working distance. Now what is this working distance means? Distance between front surface of lens and surface of cover glass or specimen. Now, if you have already operated or seen, you have seen microscope, or you have already operated normal compound microscopes or even simple microscopes in your earlier school days or college days, then you might have seen that suppose if this is the slide, okay. Now you are putting this you have kept your slide on top of the stage clip, right? Now on the stage clip you have kept your slide. On the slide there will be two things. One is either it will be completely only the slide will be there onto which your specimen will be attached. On the other hand, if it's kind of a specimen like smears, or suppose you want to prepare one permanent slide. In that case, what you will do, you will fix that particular sample with the help of a cover slip. Now when you are using a cover slip, then the working distance will be different, and if you are using only the specimen, then your working distance will be different. Now let me explain you how it will be different. Suppose if there is a cover slip onto this one, okay, if there is a cover slip here, if it is attached here, now from this one is the objective, okay, this one is the objective lens. So from here the lens is here, okay, this one is the lens. Now from here the distance that will be obtained, okay, the distance that will be obtained till the cover slip, that will be termed as the working distance. That means the distance between the objective lens and the cover slip will be taken into account. But if you are not using any kind of cover slip, then what will happen? Then the distance will be counted from the objective lens to the slide, okay. So that will be your working distance at that particular time or at that particular moment, because here in this case you are not using any kind of cover slips. Okay, I hope this will be clear for you. Now let us move into the next variable. Last one, that means the last variable of microscopy is contrast. Now I guess this contrast, the terminology contrast, is very much familiar to you all. Why? Because all of you are having smartphones nowadays. Everyone is having Android mobiles or different other kind of mobiles. So contrast is a very familiar terminology for all of us. Now, do you know what is actually contrast is? So contrast is nothing but differences in the intensity. Intensity between what? Intensity between two objects, okay, or between an object and its background. Now suppose in this case, this is an object that has been captured by a microscope. This is an image. Now if you enhance its contrast, okay, then the image will look like this. And if you are reducing the contrast, then it will be even more darker, right? So that is contrast. So that contrast will come from the intensity differences. From where it will come? It will come from the intensity differences that will be between the objects or between an object and its background. Because here this darkness will be visualized, or that can be visualized from our end. If we try to visualize them, then it will be nothing but the background that will be reflected. So the background will be, on the basis of the background, the object's intensity will be differentiated on the basis of this contrast module, okay. So this contrast is actually kept in the microscopes in order to find out the difference in the intensity between two objects, it may be, or it may be between the object and the background. So this is all about the variables that are being used in normal microscopy, and further we will have to discuss about the types of different microscope and certain common microscopes that are normally used in research laboratories. So before moving into it, let us first start with the types of microscope. So the first, actually the microscopy is mainly divided into two types. One is light microscope and the other one is electron microscope. So this light microscope, why does this term light, means from where does this term light came? Actually, is that here in this kind of microscope, light is generally used. This light can be, sunlight, or it can be even artificial lights. Okay, so there are certain microscopes, like simple microscopes are available, where you might have already used it in your previous classes, where you might have focused the light through sunlight, right? So here we will be discussing about different microscopes which uses light as the source, and the second one will be electron microscope, which uses electrons. Okay, so this electron microscope never uses sunlight or any kind of artificial light. Remember this. Now, there are different kind of light microscopes are available, like bright field microscope is there, dark field microscope, phase contrast microscope, fluorescent microscope. So these are the most common, used light microscopes in the different kind of laboratories. Electron microscope, we generally see transmission electron microscope, which in short is known as TEM, and scanning electron microscope, which is in short known as SEM. So what is bright field microscope? So always remember, there are two kind of microscopes I have already told you about. One is bright field microscope and the other one is dark field microscope. So it has been seen, or it has been mostly observed in students, that they get confused, okay, while writing in exams or while projecting it somewhere. What is the difference between actually bright and dark? So I can give you a very simple formula, or you can I can give you a very simple hint through which you can remember what is bright and never get confused with bright and dark field microscopy. So bright field, so here it is field. So as it is written here as bright field, okay. So this bright field, if you concentrate on field means the background. Okay, if we say something about field in this particular microscopy, we'll be talking about, that is, we are talking about the background. So see this terminology of field means background. So if the field is bright, okay, field is bright means here you can see through the image, the field is bright here, right? So when the field is bright, then we'll term it as bright field. That means if the background is bright, then we'll term it as bright field microscope. And what happens here it produces a dark image against the brighter background. Here you can see in this particular image that this background is completely bright, and against that a dark image is being produced, right? So in all these images, this one is A, B, C, D and E. In every all these images, you can see here that the images that are being produced are darker as compared to if you compare it with the background. So background is brighter and the objects are darker. So it has several kind of objective lenses that are used for this particular microscopy, and it has got mainly two types. So simple and compound microscopy is there. So I have already told you about the simple microscopy, where it contains a single magnifying glass. This particular microscope is taught to students, or it has been shown to students at a very preliminary stage of their schooling, mainly in the higher schooling. That means you might have come across in class 10th, 9th, 10th, 11th or 12th, okay, where you might have used this single magnifying glass microscope, which is known as simple microscope. Then the compound microscope will be there. So what is this compound microscope? Here it will contain a series of lenses for magnification. There may be different kind of series of lenses which will help you to observe a magnified image. Okay, that the light passes through the specimen into objective lens. Okay, then what will happen? The light will pass through the objective into the specimen. Into the objective lens. Oil immersion lenses also are introduced there, which helps in the increasement, or you can say it can increase the resolution of the microscope. Then it will have one or two ocular lenses. You might have observed in different labs, or particularly in case of the lab that you are following or you are coming across, you might have seen two kinds of microscopes. One will have a single lens, single ocular lens or eyepiece lens, and the other one, there will be another one where you can visualize two lenses, which will be binocular, which is also known as binocular ocular lenses. Then its resolution is almost equivalent to 200 nanometer. Okay, it will provide you a resolution of at least 200 nanometer. Next is your dark field microscope. As I have already told you about the bright field microscope, where the background or the field was brighter. Now here in this case, the background will be darker. You can see from this image that the background is darker here, whereas on the other, in case of the bright field microscope, it was brighter, and the image in the bright field microscopy was darker, but here it is lighter. Okay, so it will produce a bright image of the object against the dark background. An optical system to enhance the contrast of unstained bodies. So here, as the background is darker, so if you want to visualize an unstained body, that means if you are not staining with any kind of stains, then also you can visualize them under this particular microscope. Specimens appear gleaming bright against darker background or a dark background. So here, the specimens that you will be able to observe will be gleaming bright as compared to the other microscope, that is the bright field microscope. So next one is phase contrast microscope. This particular phase contrast microscope was first described in 1934 by a Dutch physicist Fritz Zernike. Okay, so it produces high contrast images of transparent specimens or transparent bodies. So if you are if you are having a specimen which is transparent in nature and you want to see a high contrast image of it, then what you need to do, you will have to take it for a phase contrast microscope. If you take it to the phase contrast microscope, then it will provide you a high contrast image of it. So living cells can also be examined in their natural state. Like for example, there may be bacteria which may be moving. Okay, you are having a sample where the bacteria is still moving, but you cannot visualize them in some normal microscope. So here, if you can use a phase contrast microscope, you can see the living cells which can be examined in their natural state. That means whatever in the natural state they are currently existing, in that particular natural state you will be able to visualize them. Next is, it enables visualization of internal cellular components. So it also helps you in visualization of the internal cellular composition. So internally, whatever the cellular composition is there, that also you can visualize to some extent. It helps in diagnosis of tumor cells. Okay, so tumor cell diagnosis is also done with the help of phase contrast microscopes. However, nowadays there are different kind of sophisticated microscopes are also available which are used in diagnosis of tumor cells. Then ideal for studying thin specimens. So here in this case, if you are preparing one specimen, and if it is thicker, okay, suppose if you are trying to visualize one tissue, and suppose you have made it very thicker, then what will happen? It may not be you may not be able to visualize it under this kind of a microscope. Why? Because it is very much ideal for studying thin specimens. So thin specimens, if you are using, if you can make it much thinner while preparation of the specimens, then it will be a very good choice of yours for visualization of the microscope, visualization of the images. Sorry. So let us now see about the or discuss about the principle of the phase contrast microscope. Different wavelength of light rays detect differences in color. So this theory we have already studied in our earlier classes, where we have we came across different terminologies like Gibbs-Ure, isn't it? Which where we have seen that different wavelength of light rays detect differences in the color. Different shade of gray are distinguished to our eyes due to difference in amplitude of light rays. So why different kind of shade, shade of gray? One sec. Yeah. So different shade of gray are distinguished to our eyes. That means if there are certain shades, okay, of gray shades are there, then those shades of gray can be distinguished by our eyes due to differences in amplitude of the light rays. So what is the amplitude of the light ray? According to that only it will be visualized, or you can see the differences. So this PCM or the phase contrast microscope converts invisible small phase changes. Okay. So while distinguishing or differentiating between different shade of gray, this PCM, the phase contrast microscope, what it does, it converts the invisible small phase changes. Whatever the small phase changes will take place, that small phase changes will be converted by the PCM and make it visible, okay, to you. So PCM converts invisible small phase changes caused by the cell component into visible intensity changes. So what it will do, it will convert the small phase changes that are invisible to our eyes, that will be made visible. Okay, so it will be converted into visible intensity changes, so that you will be able to visualize the cell components. Okay, cell components quite easily with the help of this microscopy. So a bookish definition is there. Like, it is an optical illumination technique in which small phase shifts in the light passing through a transparent specimen are converted into contrast changes in the image. But I will prefer to go through this for the students. Why? Because it becomes very easier for us to understand rather than going through this particular principle. Okay. So if you want to write it in exam, then you can use this particular definition. That is all right. But if you want to gain the knowledge, or if you want to gain, you want to remember how this particular microscope works actually. So in that case, it will be better if you go through this particular statement, where it is written like PCM converts invisible small shift changes caused by the cell component into visible intensity changes. Now, how does this machine or how does this microscope works? This is the light, right? So this is the source of illumination. Then there will be one structure known as the annual diaphragm. Thereafter, a condenser is there, specimen will be there. Okay. So here the light ray will be going through this, from the condenser it will reach into the specimen. Similarly, here also the same way light will be going and it will be pointing to the specimen. As soon as it reaches the specimen, thereafter it will be again refracted towards the objectives. So now it will reach to the objective. After reaching the objectives, it will again be diverted towards a plate. Okay, this particular plate, which is known as phase plate. Okay, so this will go through the phase plate, and when it reaches the phase plate, then the small phase changes will be observed, or the small phase changes will be detected by the system. So as soon as it detects the small phase changes, thereafter it will convert it into the visible intensity changes, and finally you will be able to visualize your image with the help of the objective, or nowadays different kind of softwares are available which are attached to the PC system, and then through this also you can easily visualize them. So for today, this will be the last topic that we are discussing. After this, we will be discussing about the rest of the microscopes that we have already told in the previous slides. Thank you.